Hello everyone, welcome to the topic agarose gel electrophoresis. This agarose gel electrophoresis is a basic technique that is used in all molecular biology, genetic engineering and advanced techniques that are involved in biotechnology. Especially PCR, RAPD, RFLP, fingerprinting techniques and DNA sequencing are all dependent on this basic agarose gel electrophoresis, where separation of DNA fragments is highly essential, without which the next further advanced technology cannot be carried on. This technique is used to separate DNA fragments under the influence of an electric field based on their size, shape, molecular weight. The smaller the size, the easier is the movement of the DNA fragment, and thus high resolution of the DNA fragment that is possible in this technique. Shape, when it comes to the shape of the biomolecule, generally fibrous molecules tend to be easily separated out when compared to the globular molecules. Molecular weight of the DNA fragment that is varying around 5 base pairs to 1000 kilo base pairs can be carried on through this agarose gel electrophoresis technique. It is finally achieved by moving these negatively charged molecules under the influence of electric field through this agarose matrix. The technique. This technique can be discussed in 4 phases, that is casting of the gel, loading the samples, electrophoretic run, staining and visualization of the final gel after the resolution of the bands is complete. A nice setup of agarose gel electrophoresis can be seen in this picture, where it consists of a agarose gel that is well placed in a buffer tank, samples that can be loaded into individual wells, and the entire setup connected to a power supply. Power supply of 50 to 150 volts is desirable for normal separation of the DNA fragments. Agarose, in this gel electrophoresis technique, agarose, which is a linear carbohydrate molecule or a polymer that is extracted from seaweeds, is commercially available and can be procured commercially through various companies. These sugar molecules, when mixed with a liquid substance such as buffers, can solidify and form into gels having porous internal structures that facilitate the movement of these DNA fragments. Matrix of the agarose gel acts as a molecular sieve through which these DNA fragments move under the influence of this electric field. As we see in the picture, the polymer of agarose, which consists of CH2OH side chains, can be melted into a uniform gel porous like structure using buffers in a conical flask, and then which are allowed to set at room temperature. Lower concentration of agarose, that is agarose powder mixed in huge quantities of buffers, facilitates the movement of larger DNA fragments, and thus experiments involving larger DNA fragments should go on proceeding for the lower concentration of agarose gels. If our experiments are involving smaller DNA fragments for the purpose of studying individual genes and alleles present in these genes, higher concentration of agarose can be prepared. When coming to the terms lower concentration, 0.8 to 2% are considered as lower concentrations, and any concentration between 2 to 5% is considered as a higher concentration of agarose gel. Coming in detail to the technique, casting the gel. This is the first and foremost important step, where improper casting of the gel can lead to undesirable results and also no resolution of the DNA bands. Agarose is melted mixed with buffers such as TAE, TBE and boiled in a hot air oven or a microwave oven, where a clean and uniform solution is poured. This melted agarose is now poured into a cast wherein a comb is placed for facilitating the creation of wells where we will load the samples. This gel should be completely set hard before use. Coming to the loading of the sample. Generally the samples consist of DNA or RNA or protein. Whatever be the sample, this sample has to be purified through initial steps such as centrifugation, where we get only the sample desired to be resoluted in this technique. Once this sample has been prepared, a loading buffer along with a tracking dye such as bromophenol blue is mixed with the sample and then loaded into the wells. Compounds such as phycol or glycerol are also mixed with the sample so that a higher molecular weight of this sample can settle down well in the sample wells that are prepared. Now the electrophoretic run. Electrophoresis can be generally done horizontally or vertically, wherein which most of the labs prepare horizontally casting trays and thus run agarose gel electrophoresis based on the setup. This agarose gel is now set into a electrophoretic tank or a buffer reservoir as we call, which are connected to electrodes positive and negative, that is anode and cathode, and started for the power supply. Once the power is started, it is set to 50 to 150 volts of current and thus electrophoresis is carried on. Once the gel starts running, we can see the bands running as blue colored compounds within or through the gel. These bands, once they reach 3/4 of the gel, by visualization of this blue colored bands, then it indicates that the gel run has to be stopped. Staining and visualization. Once this gel is run, DNA or RNA bands that are visible as blue colored bands should be stained with a counter dye such as ethidium bromide, Cybar, gel red, brilliant chrysal blue, Nile blue sulfate, etc. Ethidium bromide is considered as mutagenic, so alternative solutions such as Cybar green or gel red are mostly preferred. These gels now when once stained can be visualized in a UV trans illuminator or in an autoradiograph as we can see in the picture. This picture, a classic example of agarose gel electrophoresis, which has run resolved DNA bands into various sizes, can explain what actually agarose gel electrophoresis can do and help a scientist doing biotechnology experiments. As we can see, the series of bands in the ladder numbering as 500, 1000, 1500 refers to the base pairs of the DNA present in the DNA fragment, and the samples that are run in the next proceeding wells like 1, 2, 3, 4, 5, 6 correspond to the marker DNA ladder and their size that has been achieved during the resolution of this gel. So, here are few video links which are for our quick understanding and for a detailed procedure. Few textbooks such as Biophysical Chemistry by Upadhyay and Upadhyay, Gel Electrophoresis, Nucleic Acids by Robin Martin can also be referred for a better in-depth understanding of gel electrophoresis technique. Thank you.